All right, so to continue on our discussion of receptor proteins, I'm going to go ahead and switch to a different type of receptor protein. That type is a insulin receptor protein. Well, insulin is fairly specific, but the type of receptor protein is an enzyme-linked receptor. Okay, so if you think back to our GPCRs, those were proteins that would associate with a protein that would associate with an enzyme and activate an enzyme. But here what we have is a little bit different. Now, insulin is a peptide hormone that is produced by beta cells of the islets of Langerhans and pancreas. Insulin reaches target cells such as liver, muscle, or fat tissue through the bloodstream. Now, binding of insulin to the insulin receptor initiates a cascade of events that leads to increased glucose uptake and metabolism. The inability to make or sense insulin is described through the disease known as diabetes. Now, here what we have is a model that's showing a couple of different players here. Now, the first players that I'd like to kind of bring in, and I lost my mouse. There we go. There it is. Okay. Are these little hexagons here? These are glucose molecules. Now, these glucose molecules, they are aiming for what are known as glucose transporters. So these are proteins, I know that I described them in lecture, they kind of have an opening, and whenever they bind glucose, they're opening, they bind glucose, and their opening, which was previously to the extracellular space, now flips. They undergo a conformational change to move something from one side of the membrane to the other side of the membrane. Now, what this is going to show is these glucose transporters are stored within the cell in membrane vesicles. So notice how these are open, and this is kind of strange, but these, I'm going to draw as an example, this vesicle right here. On the interior of this vesicle is the same thing as what's on the outside of the cell. Okay, so all of your glucose transporters are open to the interior of that vesicle. Those vesicles will bind to and merge with the plasma membrane. And when they bind to and merge with the plasma membrane, what happens is that these vesicles go back to their natural kind of way, in that the interior of those vesicles is the extracellular space. So they will kind of merge and they'll open up. And when they open up, those specific proteins are open to receive something from the extracellular space. Now, what happens is these will bind to glucose and bring it in. But when insulin interacts with its receptor, vesicles move from the surface, and it's that fusion that actually causes this to take place. So when insulin interacts with its receptor, which is illustrated right here, vesicles move to the surface and fuse with the plasma membrane, increasing the number of glucose transporters in the plasma membrane. Okay, so these glucose transporters, they're upregulated in the presence of insulin. Now, when insulin levels drop, glucose transporters are removed from the plasma membrane through a process known as endocytosis. So this right here is an illustration of what you see whenever you don't have a lot of glucose or a lot of insulin present. These vesicles are kind of floating around. These glucose transporters are there, but they're not able to bring glucose in. So it's this insulin receptor that binds insulin that causes these vesicles to go to the plasma membrane. Now, the smaller vesicles are going to fuse with larger vesicles and make what's known as an endosome. Patches of these endosome-enriched glucose transporters bud off to become small vesicles, ready to return to the surface when insulin levels actually go up again. Oh, oopsies. So insulin binding to the extracellular domains of the receptor activates the catalytic domain inside the protein. Now, if you remember, this was an example of autocatalytic activity. So this was autophosphorylation. Now, the catalytic domain in one receptor phosphorylates tyrosine residues in another receptor. Receptor autophosphorylation allows binding and phosphorylation of protein IRS1. So what we have is this protein or insulin bind to our extracellular protein, which causes a conformational change, which causes the triply phosphorylated and the fully activated form of our protein. We have a tyrosine residue, which we've talked about tyrosines being sources or tyrosines being targeted for phosphorylation. And that's how some proteins are regulated. Think about tyrosine as a hydroxyl group up there. So the triply activated, the fully activated and the triply phosphorylated protein is going to be able to help move the vesicles to the plasma membrane. So this IRS1, and if you follow this model, what we have is insulin receptor binds insulin and undergoes autophosphorylation on its C termini tyrosine residues. So this autophosphorylation on C termini is going to attract this protein, this insulin receptor, this protein IRS1. So that the enzyme linkage is this ability to transfer a phosphate group. So when we have this part of our protein right here, fully phosphorylated, that part of our protein has a catalytic activity where it can transfer a phosphate group onto IRS1. So the binding of the insulin in the extracellular space is its receptor function. Its enzyme-linked receptor function is being able to transfer a phosphate group onto IRS1. So our insulin receptor phosphorylates IRS1 on its tyrosine residue, so that IRS1 also has tyrosine residues. Now, the SH domain of GRB2 binds to phosphorylated tyrosine of IRS1. What that is going to lead to is basically the signaling cascade. So with respect to insulin, here's a big part of what's happened. But the signaling cascade, the rest of this is, well, we already looked at insulin binding to the insulin receptor, leading to a vesicle fusing with the membrane. But then there's other stuff that's happening. This IRS1, what it's able to do is associate with all these other proteins. When all these other proteins are or have been activated by IRS1, we're going to lead to, ultimately, synthesis of new proteins. We're going to lead to both activation of transcription and translation. So indirect interaction of phosphorylated IRS1 with the protein RAS initiates a series of protein phosphorylations. ERK, one of the phosphorylated protein kinase, is able to enter the nucleus. So this is a protein that's found in the cytosol, but is able to move, when it's phosphorylated, into the nucleus. And when it's in the nucleus, it identifies a transcription factor, so a protein that's involved in activating transcription. That protein is ELK1. It becomes phosphorylated and stimulates the expression of specific genes. So it's going to lead to the expression of glucose transporters. So this kind of has like a doubling effect. Insulin binding to the insulin receptor leads to, well, vesicles fusing with the membrane, insulin or glucose receptors or glucose transporters being found at the membrane. What else does it do? Well, it leads to the production of more glucose transporters. So we have another way in which we can, or another kind of enrichment for bringing glucose into the cell. Now, the next part of this, I'm going to get into transport of ions across the membrane. I'm going to be looking at sodium and potassium channels. But these were some of the high points and examples that I wanted you to know about with respect to signaling. So your GPCRs and insulin signaling, they have different approaches, and they're going about kind of initiating transcription and translation, but they're doing it in slightly different ways. Now, both of these are huge areas of research and will continue to be for forever. I was looking the other day and I saw that I think it's like 30% of drugs that have been approved by the Food and Drug Administration in the United States target GPCR specifically, or target a GPCR. All right, well, I'll see you in the next video.